Hello. This video provides an overview of the use of whole genome sequencing in healthcare. Genetics plays a role in the etiology of virtually all health conditions, either as a direct causal factor or contributor. Although individuals are genetically quite similar, the small number of differences add up and contribute to the wide range of human disease risks. There is interest in being able to customize medical care to account for these individual differences. So what is whole genome sequencing? Well, it involves using a technique to read and record an entire human genome, which is about 3 billion letters of DNA code. It can detect variations in DNA sequence, including abnormal chromosomal structures, DNA insertions and deletions, duplications, and variations in single nucleotides. Although many variations in DNA are harmless, we know about an increasing number of genetic variants that influence health outcomes. This picture shows just the common genetic variants across our genome that are associated with health conditions, but there are also thousands of rare genetic variants which influence disease as well. So what are the health implications of whole genome sequencing? It can provide a wide range of types of health information. Some genetic variants will help explain patient symptoms that already exist. Other genetic variants are capable of accurately predicting diseases before they occur, or indicating a more mild susceptibility to developing diseases, which may or may not occur. Carrier information indicates when an individual is at increased risk for having a child with a genetic disorder, and pharmacogenomic information is used to understand a patient's likely response to different medications. The genetic information provided by whole genome sequencing can sometimes facilitate the prevention of diseases before they occur, as well as guide diagnoses and treatment decisions by clinicians. While some evidence connecting genetics to diseases is strong, in many cases the research is unclear. Therefore, the use of whole genome sequencing will evolve over time with increased knowledge. It is also possible that whole genome sequencing will identify risks that were entirely unanticipated, but that will need to be addressed. So what are some examples of whole genome sequencing in practice? Well, neurodevelopmental disorders such as intellectual disabilities, developmental delay, and autism are quite common, affecting around 3% of children. The genetic roots of these conditions are hard to diagnose since they are influenced by variants in over 1,000 known genes. In one study, whole genome sequencing was able to provide a genetic diagnosis for about half of the children with neurodevelopmental disorders, which resulted in a change in clinical care for about half of those diagnosed. Another example involves undiagnosed adult conditions. It is fairly common for clinicians to encounter patients with unusual conditions that make diagnoses and treatment difficult. Whole genome sequencing can be used to assist diagnosis of individuals that have unusual inherited conditions, sporadic disease, or extreme forms of common diseases. For example, in one clinical study, they were able to use sequence data to provide a diagnosis in 21% of cases that couldn't be diagnosed through other means. In addition to these diagnoses, they also found many unexpected genetic risks which were unrelated to the initial referral condition. So what would this look like for nurses? Initially, it is likely that whole genome sequencing will be reserved for patients with suspected genetic conditions or unusual symptoms, but it may eventually expand to be used in standard clinical care. The results tend to be complex and require expert interpretation, but they can guide prevention, diagnosis, and treatment by the care team. The sequencing results may be entered into electronic medical records, allowing guidance on different aspects of care over time. Additionally, nurses may play a role in communicating with patients about what these results mean. Thank you for listening to this discussion on whole genome sequencing. We would appreciate your perspective on the benefits and limitations of this strategy.